Now let's begin with the synthesis of this hormone. So it requires two main ingredients. One is the iodine, which enters from the blood to the follicular cell. We actually get this iodine from the food we eat. It goes into the GI tract and finally absorbed into the blood. From the blood, only 1/5 of this iodine is removed by the thyroid gland to form the thyroid hormones, but the rest of it is actually excreted through the kidneys. Now, this iodine which is present in the blood is taken up into the cell through this sodium iodide symporter. Now, this sodium iodide symporter transports two sodium ion and one iodide ion into the follicular cell from this basal membrane. The energy required for this transport is actually acquired from this sodium potassium ATPase pump. So, when the excess of the sodium is actually taken out of the cell, it uh, it maintains a concentration gradient which allows this easy diffusion of this sodium into the cell along with the iodide ion. If excess of iodine passes through the sodium iodide symporter, then the synthesis of T3 and T4 is reduced, and this is called as the Wolff-Chaikoff effect. Now this iodide ion which actually has entered the cell has to enter the colloid now across the apical membrane. So for this, the transporter here is the pendrin molecule. Now this pendrin molecule takes one chloride ion in and one iodide ion out. So this first step is called as the iodide trapping in the synthesis of the thyroid hormone. The second step is that now this iodide has to be converted into iodine. So this is done by the enzyme called the peroxidase enzyme, and this step is called the second step, that is the oxidation. Now why is this required? Because the tyrosine amino acid present within this thyroglobulin, which is the second ingredient of our thyroid hormone, it requires iodine instead of iodide. It cannot combine with the iodide ions, so it has to be converted. The thyroglobulin is the second constituent of the thyroid hormone, and it is formed with the help of this endoplasmic reticulum and Golgi apparatus, and it has a very important amino acid called the tyrosine, which is the main thing which combines with iodine. Now this is released into the colloid. Now the combination of this iodine plus the tyrosine amino acid is called as the iodination process, which is the third step. Now this iodination gives rise to two main compounds, monoiodotyrosine, which is because of one iodine plus this whole compound, and diiodotyrosine, it consists of two iodine. Now these two MIT and DIT further combine in different variations and give rise to three main things. One is T3, that is triiodothyronine, which is because of the combination of MIT plus DIT. The second one is T4, which is because of the combination of two DITs. And the third one is the reverse T3, which is nothing but the combination of DIT plus MIT respectively. Now this whole was the fourth and the final step in the synthesis, which was called as the coupling.